And with this, I would like to introduce our first speaker of the workshop, Professor Jean-Pierre Changeux, who is not only a simple speaker, he is an institution. Jean-Pierre is on the scene since 1961, and during the past half a century, Jean-Pierre produced fundamental, pioneering, and seminal work in several fields, including chemistry and neuroscience. He started his career with Monod and Jacob, the two very famous Nobel Prize winners. And in their laboratories, Jean-Pierre, in his PhD work, worked on allosteric regulations. And in his very pioneering papers in the very early 60s, he described the allosteric modulation of proteins, protein enzymes. And with his idea, a new era started in the field of biochemistry and biology. Later on, among many, many pioneering discoveries, he described the first acetylcholine nicotine receptor. And of course, he entered many different fields, published more than 600 papers and dozens of books in which he described different areas, not only in biochemistry, but he focused primarily on brain research, the human brain, human cognition, human intelligence. So he is an institution. And it is our pleasure to welcome Jean-Pierre in this room and give him the word for the next 1 hour. Thank you, Jean-Pierre.
Balázs, for this very generous introduction. I hope the institution is still alive for a while, but I was deeply touched by all what you said. And I am also extremely pleased to be with you here in Singapore since we met together with Per Roland, actually, more than 20 years ago, and have followed our work in a reciprocal manner with a great interest, and I may say I admire your work, and as also of Per, and all the brain imaging studies that you have been doing in the past decades, and all the brilliant ideas you had, and in particular that one today. And I think it is the first time I am speaking about this topic about East-West connections. And I may say this is really an extremely interesting topic. And I wish that this workshop and the whole seminar series that you plan will be very successful. And it is, of course, the right place here in Singapore and the right time to do this kind of approach. And I may say that in addition to what you said, there is a very strong issue, and I will close on that, of having a better understanding and exchange between East and West. More fraternité, I would say. And the other aspect, which I think is extremely important, is that the outcome of cognition is, of course, perhaps to talk with aliens, but also to cure disease, because there are many diseases now which are plaguing our world, which are associated with deficits in cognition that we have to understand. So all these reasons are more than important for the plan you have in mind. And I will today discuss one aspect of the cognitive sciences, which is the development of the human brain, the development of cognition, and more specifically, the development of what I have called cultural circuits in the brain, and maybe find an interpretation on the basis of a theory I proposed several years ago with Collège and Danchin. You shall see about it. Now, it's clear that there are cultural differences between West and East. There are cultural differences which deal with the oral and written language, with the symbolic and philosophical systems, which are illustrated here. One of the more important philosophers on the West are certainly Democritus and the theory of atoms. And here, Confucius and all these writings about life, about good life, I would say the homologue of Aristotle at that time. Now there are clearly differences in writings that you can see here, and of course of artistic style. And I must say I'm pleased to show this slide to illustrate, I would say, the high standards on both sides. I'm not going to select one against the other. All of them are of great interest, and we have really to go further in understanding these differences and benefit from the understanding of these differences at the level of the human brain. Now, to further understand cultural diversity, we have to develop an analysis at several levels of organization. And this is something very important for us as far as the human brain is concerned. We have to realize that the human brain has to be understood at different levels, at the gene level. First of all, we have to understand what are the species-specific universal dispositions for cultural acquisition in human brain anatomy, physiology, cognitive function, conscious access. We have to understand the epigenetic variability which is taking place, which gives rise to faster evolution than the genetic evolution, which is involved in the origin of the human species. Then we have to understand also access to consciousness for testing our internal with the external world. And the genesis of what I call epigenesis rules, you shall have a definition later on. And then we have to be concerned by the issue of social groups, differentiation and survival. And of course, of some conventions which are contingent to a given society, but also other ones which may be more universal, and in particular, scientific knowledge, and why not at the global scale, the human rights. So I will start with the first, the universality of the human brain at the genome level. Now we have to realize that we have brains, all of us, which are made of billions, 87 billion perhaps or more. Nerve cells, which are interconnected through a very complex network, which is discontinuous, as Harmonica had mentioned. And the number of these discontinuous contacts or synapses is in the range of 10 to the 15. And the signals which circulate in these pathways are either electrical or chemical. And in both cases, they can be understood in molecular terms. And the synapses are the places where the chemical signals are released, the neurotransmitters, by the nerve impulse, cross the boundary between the two nerve cells, meet with the receptors which are present in the postsynaptic membrane, cause a new electrical wave which gives rise to a nerve impulse. So these different steps now can be analyzed at the molecular level. I would even say at the atomic level. We know the atomic structure of one of these, and now several of these receptors for neurotransmitters. This is the receptor for acetylcholine, which is the first identified. But now there are 3 or 4 more which are known at the atomic level. We even know that the signal transduction, which is mediated by these receptors, can be understood again in the microsecond time scale. And these conformational change, in fact, are rate-limiting steps from information processing in the whole brain. You may have asked yourself why the brain is so slow compared to a computer. Why is not working with electromagnetic waves, but with propagation of impulses which are slower than the speed of sound? Well, there are rate-limiting steps, and these rate-limiting steps are the ion channel and neurotransmitter receptors, which impose some constraint to the whole brain. And therefore, we have to be aware of these elementary building blocks, even if we want to understand the cognitive function of the brain, because these cognitive functions are grounded on these molecular processes. And yet, our brain is not a bag of molecules. It is a highly organized system of molecules, but these molecules are not just randomly distributed in the whole brain. And where does this organization of the brain comes from? As I said already, it comes from the biological evolution at the level of the genome, and took millions of years to develop, for instance, from Homo habilis to Homo sapiens. Then there is another nested evolution, which is taking place at the level of the individual, from the fertilized egg until the adult brain. And then there are some critical steps, I will come back on them, which are crucial, because I am going to organize my talk around this. There are processes of synapse formation, stabilization, and selection, which are taking place, which introduce some epigenetic variability within the human brain organization, which is framed by the genetic program. Then there is, in the brain throughout its development, change of synaptic efficacies, which is able to introduce, if I may say, the dynamic of thought. And then there is a social and cultural evolution, which also involve the same kind of synaptic efficacies, but also extracerebral memories through which an interaction may take place during the whole development of the human brain. So therefore, we have to be clear on this. If we really want to understand cognition and cultural differences, we have to go through all these different levels all together, from the molecular, from the cellular, the circuits of circuits, and the global architectures of the human brain, which led possible the emergence of consciousness and social life. And I would be what's characterized the human brain is that it is both rational and social. And this is something that we have to deal with, and which is unique to the human brain and not to other species. And also, as I said here, we have a system which is open, because of its spontaneous activity, I will come back on that, is exploratory, which is self-organizing, conscious, and engaged in social communication and cultural transmission. All these things have to be dealt with if we want to progress on cognitive sciences. Now, the molecular biology may look simple. It is not. And to my opinion, is still not enough understood by molecular biologists, which are and feel more concerned by cardiovascular, liver, immunological, or whatever kind of disease, and are not concerned by the human brain, unfortunately. And the molecular biology paradox is the following. If you compare the size of the genome or the number of genes and the complexity of the brain, you can see that from the yeast to the fly, you have a doubling of the number of genes, which is okay, maybe. Now, you go from the fly to the mouse brain, you have maybe another doubling. Now, when you go from the mouse to the human, it doesn't change. And yet, the number of nerve cells as an indicator of complexity, because it's not, of course, enough to indicate the complexity to be an index of complexity, you realize that the number of neurons goes from 40 million to 100 billions. So how do you account for that? And I think this is the most pressing issue that molecular biologists have to deal with for the present time. And the differences in sequence, let's say, between chimps and men, without speaking between mouse and humans, is extremely small. And yet the brain of a chimp is not the brain of a human being. In particular, they don't speak and they don't write. So we have something which is added to the chimp or lower vertebrate brain through minor changes of the genetic information in the genome. And I may say that this is not solved. The recent studies of Svante Pääbo, for instance, on the genetic of the ancestor of humans like Neanderthal and others, illustrate that there are a few genes which change, but not that much. And it means that you have some mechanism of non-linearity which is involved between genetic evolution and the complexity of the brain. And as an example of the difficulty we are facing, here are the data which have been obtained and started by Thomas Bourgeron at the Pasteur Institute, actually in my department. He discovered that there are predisposition genes to autism or Asperger syndrome, and that there are maybe about 200 predisposition genes which are being identified. He identified the first one and showed that they were, in fact, genes affecting proteins from the synapse. But the finding he made was that there is not a simple relationship between genes and brain phenotype, despite the fact that many of these genes give the same phenotype, which is, again, a big paradox. And the second thing, which is that there is necessarily strong interaction between these genes so that they give, I may say, the same phenotype. So the interpretation that one can propose is that they are, in fact, assemblies of genes, as they are assemblies of neurons, which would code for assemblies of neurons. And therefore, that we have to further, this is, if you wish, some kind of theory that I have developed with Igor Tsigelny from UCSD in San Diego, which may have to be taken into consideration to try to progress in this relationship. But nevertheless, we are, if I may say, facing one of the biggest mystery, if I quotation mark, of the relationship between genome and brain phenotype. And this has to be further studied in the next decades, since we are talking about a program for the future. And the other interesting thing as far as the East-West differences is, are there in this respect, since the population have evolved separately for centuries, are there genetic evolutions which took place in the recent years? Maybe, maybe not. Are there differences between East and West about these predisposition genes to disease? All this is extremely interesting and may be a source of studies for the future to better understand these diseases, which are actually common, of course, between East and West, but maybe the ratio of the frequency of the gene may be different, and could be interesting to further analyze. Now, we have now in hands the human genome. We can do hundreds or thousands of human genome analysis. And we have also, thanks to brain imaging, ways to look at the human brain during development. And this is one of the best pictures I like from Ghislaine Dehaene-Lambert. She's the wife of Stanislas Dehaene, and which shows you the newborn brain. And first of all, you can see that there are long-distance connections. I will come back on that, which are already present there using tractography. Of course, they are not as developed as they are in the adult brain, of course not, but they exist already. And also, you can see that the baby or the newborn respond to speech. You can see that there are activation taking place in the language areas. But interestingly, the newborn distinguish forward speech that we understand and backward speech, which is the tape put in the opposite direction, that we don't understand. And interestingly, the newborn already makes the distinction between the two. It means that there are learning processes which are taking place early on, before birth. So there are universal innate dispositions for, let's say, grasping and breast sucking, for also distinguishing living and inanimate species, the mother from the father very soon. Well, there is an innate disposition for recognition of human face, and very quickly between different human faces. There is also some predisposition to conscious access. Steve Quider in Paris has already shown together with Ghislaine Dehaene that there are some signs of conscious access already in the newborn, empathy and sympathy, and here, perhaps, already some disposition to language. But, of course, these innate dispositions are not going to answer the issue that we want to deal with, which are, if I may say in short, the neural bases of cultural diversity. And we have to deal with something that sometimes the molecular biologists, I belong to them, don't understand, is that there is a difference between the liver and the brain. And what is this difference? The connectivity level. There is a level of complexity which is much more important even than anything else for the understanding of the brain, because new properties are appearing at this level, and that's what I am going to illustrate. And the epigenesis I want to talk about is linked with this level of complexity, which is that of connectivity. And from the start, I would say, is different that what is called epigenetic modification of chromatin, which came later, after our proposal, and which deal with the level of the nuclei and just take the brain as if it was a liver. In our case, it's not the situation. And I may say that there are good reasons why. And the first is if you compare the human brain at birth and in the adult, you can see that the volume and size of the brain is increasing by a factor of 5. And with Hugo Lagerkrantz, we try to count the number of synapses per second. Well, it's easy to do. We take the total number of synapses in human brain and divide by the number of seconds it took to have a full development of human brain, which is about 15 years. And you find an average number, which is 10 million synapses per second. So you look at your child 1 second, 10 million synapses, which is something extraordinary that we never realized like that. And you can see this taking place during the postnatal development of the human child. And at least 50% or more of the synapses of the connectivity of the adult brain is taking place during postnatal development. Now, this is the reason why we thought that there should be some effects of the environment on the development of the brain connectivity taking place during this period of development, which, as I said, lasts about 15 years. And together with Antoine Danchin and Philippe Courrèges, we proposed this theory of epigenesis of neuronal network by selective stabilization of synapse that we formulated in mathematical terms at the time. And the idea is very simple, that the establishment of the connection in the human brain during development does not take place like the assembly of a computer. You are not putting pieces together and link them together. We have, in fact, a development which is taking place within the genetic envelope. I emphasize that. There is some variability due to the outgrowth of the connections. There is not necessarily a one-to-one relationship established at the beginning. In this respect, the synaptic is very misleading for us. And in fact, it's the opposite. There is some random components in the way the contacts are being established. There is some kind of exuberance which is taking place. And then the state of activity of the network is contributing to the selection of some of the synapses. Some are stabilized and others are eliminated or pruned. So this is this unit of selection which I describe here. But of course, during 15 years, you may imagine how many of these schemes are nested one with the other and how complex is the whole process during development. But the main idea is there that we may link, in fact, the elementary biochemical property for synapse selection and the development of the circuits which are contributing to the building up of the brain and its cognitive functions. And here you can see the evolution of the number of synapses after birth in the rat. It's very fast. In the cat, it's slower. In the macaque, it's even slower. And the human is very long, as I said, about 15 years. And the most interesting thing is that you can see that the absolute, this is from the Unterlöscher work, that the absolute number of synapses is reaching a maximum and then goes down at the time of puberty and in the adult and, of course, during aging. So there is indeed a global elimination of synapses which is taking place during evolution. Of course, it's the envelope of the number of synapses which is taking place due to these successive selection steps which are taking place. And our view is that these phenomena are not only associated with the acquisition of basic knowledge, but also to the genesis and internalization of culture. And an interesting aspect of this theory, which we propose as a theorem, a mathematical theorem with Collège and Danchin, we stated that way, that different learning inputs may produce different connective organizations and neuronal functioning abilities, but the same behavioral abilities. It means that, in short, having this kind of Darwinian selection taking place, we may, and having a selection which is based on function, we may have the same input-output relationship, yet with variable connectivities. And this is very important because it has implication about the connectome, which has been developing in the United States in particular enormously, where people thought that one should be able to have a connectomic description of any part of the human brain. And I say, globally, yes, but in detail, certainly not, because of this particular variability due to the development of the human brain. And the proof of this was proposed by Cy Levinthal many years ago. And here he studied two individuals, isogenic of Pecilia formosa, which is a fish, and Muller cells. And you can see that, first of all, there is a variability from right to left, and also from one isogenic individual to another. So they are identical twins with different connectivity. So this is something very fundamental that we have to understand, that there is, as a consequence of the mode of formation of the connections, some epigenetic variability between individuals and even between right and left. And yet, and this is something very important, we may still have constancy of behavior. We can understand ourselves even if we know that we have physically different brains. This is a second paradox that I may emphasize for cognition, is to try to understand the invariance of our mental representation despite this variability of the human brain. Another demonstration of this variability from the work of Lichtman, which is recent. He compared the left and right neuromuscular innervation of the interscutularis muscle in rats. So these are, in fact, identical connectivities from the left and right, and you can see how variable it is here. And nevertheless, the function between right and left is the same. This is a real demonstration of the variability of the connectivity despite the invariance of the function. Look at the number of branches. You see the main branches are the same, but the details are very different. This is a beautiful work, and I think this may be generalized to the whole connectivity of the brain, of course. Now, having this in mind, one can also try to have a demonstration at the synaptic level. These are all demonstrations I did with the neuromuscular junction here, and show that one can stimulate in the embryo the activity of the spinal cord and stimulate selection by enhanced selection by increased electrical stimulation, and if you block activity, you have much less selection which is taking place. We can even produce some kind of biochemical model for this selection. And this is just a work from another group, from Stryker and Schatz, which shows clearly the pruning which is taking place when the system, this is a visual system, is active. And you can even realize that when you block the activity by tetrodotoxin, you have a much more diverse connectivity and many more synaptic contacts which persist in the adult. So this has been re-examined recently. So these were old views which have been confirmed recently and are still very, very true, if I may say. Where in the case of developing tadpole, you have exactly the same phenomenon as the one I described before. Here, when you have synchronous stimulation between pre and post synaptic ending in the eye, you can see that you have a nice selection which is taking place and very active connection, while on the opposite, when you have asynchrony, you have many more axonal branches and very less efficient connections. So there are many evidence now for this synapse selection mechanism, and I said that one could imagine a lot of mechanism at the molecular level for the selection, like here, the diffusion of receptors, or allosteric transition, which are linking the NMDA receptor with the D1 receptor. Beautiful work by Anita Peria from Stockholm. And these are some of the many molecular mechanisms which can be explored to investigate at the synaptic and molecular level this molecular basis of synapse selection, which then play a crucial role in the acquisition of what I called cultural circuits. And we have here the best example one can find, which was discovered in France by Jules and Auguste Desgerines, where Desgerines, looking at patients, was able to show that a lesion located here, for instance, can give an alexia without agraphia, which means that this lesion introduces a loss of reading without a loss of writing. The subject can write and cannot read. Of course, he cannot read what he has written, but that's for sure. The main idea from that is that, as you know, writing and drawing are acquired. They were not innate dispositions in the human brain. They have been learned historically at one stage in the history of human beings. In cave drawings around 30,000, there are already signs. There are incised tokens in the Middle East around 9,000, and pictograph in the same place at Ur in 4,000, whatever. This has been an acquisition of human culture. And therefore, there are, we have to say that there are connections which are being established during development, which create a link between vision and cognition, and have to be understood. And this is, again, the observation that Desgerines did in the case of alexia without agraphia, which is not limited, as you can see, to the visual areas, not at all. And therefore, there have been imaging studies done to look at these written language circuits, and one of the best one is by Castro Caldas and the Swedish group of Martin Ingvar. And as you can see here, there is very significant difference using brain imaging between the connectivity in the prefrontal cortex in literate versus illiterate. And there are also other differences in the more occipital regions. This group of Price and others also found differences in the parieto-occipital territories, and most of all the recent work of Stanislas Dehaene, Laurent Cohen, and others show that there are striking differences in the global connectivity of the brain, which is being established for learning and of reading and writing written language, and there are what I would call some epigenetic appropriation of neural circuits which are taking place during language acquisition, written language acquisition, which is taking place, let's say, around 5 years old. And you can see that this is not taking place in one step. And I look at my grandson carefully learning written language. It took at least 1 year for the child, through trial and error all the time. So it means that we have, of course, innate disposition to learn, but not to learn specifically the language, and to have a selection of the circuit which is taking place there take a lot of time, so that the opportunity of the connections happen when the child is learning. And here I just illustrate also something which may be further developed by others, that in the case of Japanese there is kanji and kana, and that brain imaging studies is one among many other studies which illustrates using functional MRI that there are different circuits which are being mobilized by reading kanji and reading kana. So we think that there are selection mechanisms which are taking place during not only oral language but written language at a critical stage of development in the child. And the issue is, of course, to try to understand how this link is being established between spoken and written language. And as you know, there is first a relationship between the Saussure languages line between the acoustic image, the signified, which is, for instance, tree or arbor, and the concept, which is what is meant by arbor, by tree. So the relationship between, I would say, the neural basis of meaning and the sound, which creates the linguistic sign in the case of the spoken language. Now, in the written language, it's even more complex because you have to establish a link between some kind of sign, written sign, which is linked to the sound and the meaning. So it means that in the case of written language, we have mobilization of not only the visual system, but the auditory system, which is being mobilized together with what is stored in the brain for meaning. And therefore, we have to try to understand the link between what are these social conventions, which are quite arbitrary, if I may say, and the meaning of the word in the brain within the social group. And I may just show you this picture of Titien to try to approach this mechanism, which is still, of course, to be further understood, which is a beautiful painting from Tiziano in the Louvre, the Virgin Anne and the child. And what you can see here is very interesting, is that the child look at the rabbit, And the mother look at the child looking at the rabbit. And there is only something which is missing from Titien. He did not put the soundtrack there. And the soundtrack would be rabbit, of course, when the child look at the rabbit. And this is just to illustrate, of course, the way one may view this arbitrary link between sound and meaning, and between sound, meaning, and pictures of the written language. And I may just mention the work of Thomas Hello, the issue of joint attention, which is being taken between the mother and the child. And through the joint attention, the issue of shared reward, which may take place and create a mechanism of selection of the right connection between sound and meaning. But these may be further discussed. I developed a formal model for that, for synapse selection and reward. I'm not going to show it. I have not enough time, but just to illustrate some recent developments of research on the issue of parental care, which is not exactly what I just mentioned, but which is related to it. And you can see that here one can distinguish the areas associated with parental care in rodents, which can be identified here by the group of Catherine Dulac, and recently in the human brain. Also the paternal role, which I wish to emphasize, not only the maternal one. And here you can see some illustration of the VTA activation in fathers viewing their own child compared to a different child. And here the anterior ancilla activation for fathers listening their own child cry compared to a different child cry. So there are clearly ways to approach these issues of reward in cultural transmission. Now, I may switch to cognition. I have still, what, 5-10 minutes? How many? 10? It's okay. Thank you. I can finish. And the question, of course, is access to consciousness. This is something which have been developed with Stanislas Dehaene for more than 10 years now. And I just want to show two slides on that, not to further develop, because I know there is a talk on access to consciousness. And what I want to say is just that to have conscious access, we need to have a space in the brain. This was proposed by Bars many years ago, where we have simultaneously different sensory modalities. We have sight, I see you. We have sound, I listen to you. We have the reference to our own knowledge, since I am thinking about what I am talking about, and so on and so forth. So if you have this kind of space which is multimodal, you need a way to interconnect the areas of the brain which are specialized in these different modalities. It's a very simple kind of rationale. Certainly wrong, because it's too simple, but at least it exists as a working hypothesis. And the idea, therefore, we propose is that there is in the brain a long-range connectivity which is taking place, and which was already so demonstrated by Ramon y Cajal, these pyramidal cells from layer 2 and 3, already by Desjardins, who showed this beautiful white matter long-range connection in the brain, and recently confirmed by the group of Pugliese using tractography. These neurons are located in the prefrontal cortex and in the parieto-temporal areas and cingulate cortex. Thus, the circuits, which was proposed actually for working memory by Patricia Goldman-Rakic in the monkey, between prefrontal, parieto-temporal, cingulate cortex. And the proposal is that this long-range connectivity contributes, that's the only thing I want to say, contribute to the conscious workspace in our brain. And the evidence for that is accumulating now. This is an experiment that Stanislas Dehaene has done, where he was able to develop this kind of masking experiment, which is a long standing kind of experiment. And he used it to do some brain imaging studies. And in this study, you have a succession of slides. And here, the word lion between two empty slides. And here, the word note between two masking pictures. You ask the subject here, he has not seen anything. You ask the subject here, he has seen. He can say and report, I have seen lion. And you can see that in the two cases, there are different brain images. And this is corresponding more or less to the global neuronal workspace I just illustrated before. So these are only two slides I showed on many studies, and this has been reviewed by the two of us in Neuron, you may find in 2011. But just to establish the east-west connection, all of you know meditation, which is an oriental technique, which is used to control, to self-control emotion and so on. And the group of Anslu from Denmark, you may know, has been working on that. At the beginning, I was not believing it. I may say I was a little bit suspicious. I'd say, well, but I have been more and more convinced, as you shall see. And there are clear differences in the brain before and during meditation using magnetoencephalography. And interestingly, there is a prefrontal cingulate pathway, which is being used. I mentioned the global neuronal workspace. And the circuit of self-reference has been proposed by Anslu and colleagues. Now, I thought, from a discussion with him, that if there was some kind of self-reference circuit, then during addiction, which is a loss of control, as you were, there should be a change in this circuit. And that's where I started to be convinced, really. And we did some studies together with pathological gamblers, because in Denmark, to study nicotine addiction is forbidden in humans. Well, there are rules, ethical rules that have to be followed, which is natural. I am not criticizing that. I think it's but so with gambler it's easier, and there are clear differences in these MEG that you can find in pathological gamblers compared to subjects which have not this addiction, and spontaneously and during playing a game, as you can see, which is very striking. So there is the issue of the relationship between the self to the other, which can be studied. And this is, of course, for me to conclude on that now. The relationship of one self as another, which is, if I may say, the basis of ethics in our societies. And there are briefly evidence for empathy for suffering, where the brain images are compared between a subject which is suffering from an electric shock and the brain of somebody who is looking at somebody suffering from the electric shock. So it's the self and the other. And there are some overlapping circuits. Now there is, of course, violence, unfortunately, as you know, sociopathy, which may be due to some kind of innate disposition, but most often acquired during difficulties during development and so on and so forth. And here again, one may define some neural circuits for sympathy, which means that there might be, this is a working hypothesis, again, we are here at a workshop for future research, that there might be some universal circuits for moral judgment. Now, of course, there are differences in different societies. And the issue is that there might be a way to, in fact, within this universal disposition, to orientate behavior according to a given tradition. And that's what I call an epigenetic rule, which is an acquired pattern of connections, which may be stored in the prefrontal cortex, and which limits, constrain, and organize behavior, like reasoning and computation, moral regulation, or even artistic creation. This has been studied in France by Koechlin and Disposito in the United States, and there are quite a number of studies on that going on, which are, of course, something to further understand if we want to progress in the understanding of the East-West relationship. Now, almost my last slide. We look at epigenetic rules through East and West. And quite interestingly, at nearly the same stage, same years, epoch, we find the same epigenetic rule. The golden rule can be found in Confucius: do not do to others what you do not wish done to you. Even Mo Tzu: he who loves others will be loved in his turn. He who causes others to profit will profit in his turn. Of course, in the Occidental tradition, we find it in the Bible: love your neighbor as you love yourself. And in the New Testament, do unto others as you would have them do unto you. So it means that there are norms of reciprocity in the social group which develop, if I may say, with civilization, and which are common to East and West, despite differences I just illustrated before. And there is a universal rule of reciprocity, which is, if I may say, an extension of sympathy, which I may say is forgotten in some region of the world today. And that's the reason why I will close on the Universal Declaration of Human Rights, which is something we have to think about from East and West all over the world, and in particular in the Middle East. And remember that we may think about a universal ethics through East and West. And this is just finished on Keith Haring, and just say that we have a plausible research program to study the genesis of cultures, their storage in the brain, and their transmission through oral and written language. Thank you very much. Sorry, I was a little bit long.
No problem. No questions, and I think we can go on for another 10 minutes. That is with questions. Please take a seat.
Okay.
Yeah, better.
So I very much liked your talk, and in particular, the main issue that science and therefore also neurocognitive sciences will progress from understanding these major paradoxes that we are faced with. And one of them was the fact that the computers are about, shall we say, 10 orders or 5 orders of magnitude faster than the human brain, yet they have difficulties recognizing items. So what do you think is the solution to this paradox? The brain is slow, but it is very efficient. How can it be so efficient?
That's exactly the question we have to ask if we want to build more intelligent, quotation mark, computers, which have not been done up to now. Is the reason is that the human brain in particular is highly parallel and is doing a lot of computation in parallel instead of doing them sequentially as the way the standard computers do. So having these heavy parallel systems and interconnected, being parallel as well, you know, with different levels of organization, and also a very long time scale of acquisition of meanings and of rules and different programs coming from the outside world, which the computer does not do. No, you know, the computer is not really learning knowledge, it's simply communicating knowledge. And when you ask for the implementation of meaning in a computer, they say, well, we don't implement it. You know, in the brain, we do implement through synapse patterns what a meaning is. I'm sorry, I could not discuss all these things, but I went very quickly on that. But this is, of course, something that is very critical. And we acquire this knowledge about the world, about ourselves, about the society, about the culture, during very long periods of time. And well, when I said that to my colleague in the HBP, making neuromorphic brains, you know, with artificial, say, oh, but we are having units which are 1,000 or million times faster than the neuron. I say, yes, but that's not the point. I agree that you may go faster there, but then you have to organize this device in a way that you don't when you build your computers. So I think there is an enormous progress to anticipate if we really understand your question, how does the human brain process things with a very slow speed, but a very sophisticated architecture. And we have also to progress in the learning process itself, you know, how it happens. And how it happens despite the variability of the structure. You see, which is a problem that the computer people don't have. And I think this means that there is some kind of invariance in the patterns of activity despite the variability of the connectivity. And I have some ideas about that. I think we have the theorem, which is okay, but it's not enough. And I think we anticipate a very fundamental progress there. You know, I think the people are saying, oh, I am trying to get the neural code. You know, everybody talks about, well, I think to me it doesn't mean much. It's not the neural code, it's the way we actually understand this kind of paradox. Yes, yes, there is a microphone for you.
Can the computer be an enabler to enhance the operation and the efficiency of the brain?
A computer enhance.
Be an enabler that empowers the brain to work more efficiently.
But why do you want to have the brain work more efficiently? You are not pleased by your brain?
No, if let's say if we were to deal with, let's say, students,
To
right?
have students faster and brighter? Why don't you let them develop normally?
But if let's say you have a computer that can enhance the operation well, they can understand the issues better, they can what you call analyze better, and they come up with a better impact of learning.
But you do that when you have a computer. When the normal subject is using a very sophisticated computer, why do you want to have a computer which is going to modify your brain? You know, this is the issue of enhancement. I think it's an ethical issue. The question you asked, if I understood well. The issue is that you want to have your children learn better and faster. That's the issue?
It's not learning better of what you call faster, but create a clarity of understanding, you know, that can
A
help.
more intelligent brain.
Well, in some ways, yeah.
Yes, but I think each time one has tried to touch on the human brain, as you know, through genes, through drugs, through anything like that, you may enhance some of the disposition, but alter other ones. So I would be very careful in doing that, you know, because you may have a child who learns faster, but who is not going to recognize his mother and his father, or who is going to have very aberrant behavior with his colleagues at school and so on and so forth. I would be, as far as I'm concerned, extremely careful about these studies. I think the issue for the development of children is to have them develop as we are, but in a much better way than we do through social interaction between human beings before doing it with computers. Now, I don't want to say that we cannot improve the teaching through computer, which is perhaps what you said. I think we may, but we have to be very careful about that. You know, because our brain has a lot of plasticity, which can be positive, as you said, but it also can be negative. And the problem is that we have a very complex system which is working in a global way. And when you touch one part, you may have other effects and different and negative effects on other parts of the brain. So I would be very careful. We can further discuss this issue because I think it's not only a scientific issue, but it's also an ethical issue. And I know that there are some philosophies, even by very distinguished colleagues and scientists, who say, oh, we have to give the drugs, we have to modify the genes, we have to, and so on and so forth, so that we have a more intelligent human being. Well, I am not sharing this view. Maybe some of you do.
No, I don't.
But I think the main issue is how to find a good way of utilizing what we have in our genome, you know, just spontaneously, to develop a human brain which is equilibrated, which is social and rational, and has a way to behave normally. You know, that's for me the more the real problem. And in fact, if you look around the world, this is not the case. You have very poor countries where the children are developing in a very bad way. Nobody is taking care of them. And even in our Occidental societies, Wolf Singer has shown that at the time, I did not discuss that, I could have another talk. When you have at the time of puberty, there is a dramatic loss of EEG coherence, you know, the one I illustrated in the case of access to consciousness, whatever, by Meg and so on. And there is a dramatic loss. It means that adolescents at between 14 and 16 years old, there is a dramatic reorganization of the brain which is taking place. We know that from them and from other studies. What does the society does? You know, they are either taking drugs, being addicted to computers, and fighting in the street, being recruited by the Islamic State or whatever this time. So, you know, we have to realize that even in our so-called civilized societies, we are not always taking care of the education of the children the way we should. And also, one aspect which I did not discuss, which is very important, is what I call secular education, in the sense that the children should have the main ethical rules which are being taught very early on. And now the issue of beliefs is a family problem. It's to deal with it with the family, and at school the teaching should be much more universal. That's a long French tradition, as you know.
One short last question.
All right, so put it shortly. How redundant and how plastic is the representation of consciousness in our brain?
You know, the issue of the physiology of conscious representation versus non-conscious is a very lively kind of issue. It's being studied. We have some views, but we thought with Stanislas that conscious access is manifested by what we call an ignition of the global workspace. And we should further specify this, because the question is whether in this ignition there is a redundancy of the initial information throughout the whole brain, or something else which is taking place. We don't know. And this has to be worked out. I think it's a very important question. Maybe we are going to talk about it.
Apologies. We would love to go further on, but we have a very tight schedule. So may I propose that we stop here? Anybody who has questions to Professor Jean-Pierre Changeux can give him questions in the break. And we have to continue at 12:30, but I think we give 5 more minutes. So 10:35, we meet here again, okay? Thank you, and one more applause.